All right, hello everybody. It's Mr. Brott. I know I haven't seen you guys for a while. I hope everything is going well. What I wanted to do was just put together three separate little videos, kind of reviewing each of the three chapters for uh, nervous system, um, starting with chapter 47, animal development. So we'll do a little video on each chapter here, and hopefully this will kind of help you guys out as you're reviewing and studying for the test for tomorrow. Um, So let's start with chapter 47, looking at animal development. I'm going to kind of jump down through the slides here a little bit and kind of hit on the main points. But starting out with animal development, one of the main things that you want to remember is this embryonic development stage of a new organism starts with obviously fertilization, sperm and egg fusion, which creates that zygote, that single cell, followed by cleavage, which really just means division of those cells. So going through mitosis, gastrulation, which is going to be the production of cavities, starting in that what's called a blastocyst and a gastrula, where the cavities start to form within that ball of cells, and then differentiation starts to occur during organogenesis, before that new organism is born, either live or through an egg. Okay. All right. So then, starting down here, whoops, uh, looking at the difference between determination and differentiation. So cytoplasmic determinants, which are going to be basically signaling molecules within the cytoplasm, are going to help decide where or what cells become what specific types. So determination is the term used to describe the process by which a cell or group of cells becomes committed to a particular fate. Differentiation is going to refer to the resulting specialization in those cells, in their structure and function. So obviously cells in a multicellular organism are going to share the same genome. Every cell is going to have the same exact DNA. So the question then is, well, how is your skin cell different from your brain cell? Because they obviously have a different function. Well, it all goes back to which genes are turned on and which genes are turned off in those particular cells. So it's the expression of those different sets of genes. We have something called fate mapping, which is a diagram that shows organs and other structures that arise from each region of an embryo. Okay, so looking at this one, a fate map of a frog, for example, we have the blue cells that are the epidermis. That's going to form an outer layer of that frog. So for humans, it would be like our skin. You have the central nervous system, which is in that kind of teal color. You have the mesoderm cells, which make up kind of the middle. Mesoderm you can think of as middle cells of that frog. So those are going to make up some of the internal organs and muscles. And then the endoderm is going to be cells that usually line your cavities and are on the outside of some of your organs. Okay, axis formation. Now when you're thinking about how an organism develops in three-dimensional space, so what cells go on the front, what cells go on the back. If you think about a human being, why is this organ in front of that organ? Why isn't it the other way around? Things like that. So a body plan with bilateral symmetry is going to be found across a wide range of animals. A body plan with asymmetry across the dorsal ventral and anterior posterior axes is going to be non-symmetrical. The right and left axis is going to be largely symmetrical. So we can look at some examples of that too. So if we think about, let's see, let's skip that one. So what I wanted to get to is looking at what do these words up here mean. Okay, dorsal, ventral, anterior, posterior, and then you have obviously right and left. which have some other special names, but dorsal is going to be the back side. Ventral is going to be the front side. Anterior, the top. Posterior, the bottom or the back. And then you have what's called medial and lateral. Medial means closer to the midline. Lateral means further away from the midline. Okay, I'm going to swing down here to formation of the vertebrate limb. So we know that a lot of vertebrates have limbs, as we do, 2 legs, 2 arms. Inductive signals are going to play a major role in pattern formation, development of spatial organization in these organisms. So the molecular cues that control pattern formation are going to be called positional information. This information is going to tell a cell where it is with respect to the body's axis. So where is that cell in 3-dimensional space? Which direction should that cell face? Not only in the position where it's at, but in relation to other cells around it. Oops. And then it determines also how the cell and its descendants respond to future molecular signals as well. So if there was damage to a specific organ and your body is repairing it, it wants to make sure it repairs it in the right way so it looks exactly the same as it did previous. So the wings and legs of chicks, like all vertebrate limbs, begin as bumps of tissue called limb buds, which we have as well. So if you want to see a picture of a limb bud, that's what that looks like here. That little bump that comes out. So the embryonic cells in a limb bud respond to positional information, indicating location along 3 axes. So proximal versus distal. Proximal means closer to the midline, whereas distal or distance, further away from the midline. So for example, your shoulder is more proximal compared to your wrist, which is more distal. Anterior, posterior. Anterior means the top, which would be like your head. Posterior would be your bottom, like your bottom. Dorsal would be on your back. Ventral would be on the front. One limb bud regulating region is called the apical ectodermal ridge, the AER. It's thickened ectoderm at the bud's tip. The second region is the zone of polarizing activity, the ZPA. That's a mesoderm layer. You remember you have epidermis on the outside, mesoderm underneath that, which is under the ectoderm, where the posterior side of the bud is attached to the body. So for example, tissue transplantation experiments support the hypothesis that the ZPA produces an inductive signal that conveys positional information indicating where posterior is in that 3-dimensional space. So as the limb bud develops, it's going to grow outward away from the posterior side of that organism, so that it develops out away from it. It becomes an actual limb, and it doesn't start to grow inward instead. All right, so that was kind of the main basics about chapter 47 and animal development. We just kind of hit on the main parts.